Swayam Prabha. Digital India. Educated India.
Hello and welcome to our today's lecture of Introduction to Mechanobiology. So, in last lecture we had started this alternate mode of migration called amoeboidal migration, amoeboidal motility. Okay. So, in compared to mesenchymal migration, so I am just writing MM. So, this is AM. So, MM, where it is adhesion dependent, in AM you have lot more shape changes, okay. And adhesion amoeboidal migration is characterized with cell speed. So, VAM is typically much greater than VMM. So, these can migrate in amoeboidal motility, cells might migrate 100 times faster than mesenchymal cells. Okay. So, then I said that what are the different modes of amoeboidal motility, and in that regard, so in 2D I said that there are 3 different possibilities. You might have ACP, where just like mesenchymal migration, cells protrude, sends actin based protrusions, stabilize the protrusions and use contractility to pull the cell forward. Okay. So, this is just 2D normal mesenchymal migration. You can have 2D protrusive, okay, where your protrusion force is much significant and adhesion is there and contractility is insignificant. Okay. And this is possible because these cells are small in size. If the cell was huge, then this kind of localized protrusion will not lead to accumulation of membrane tension. As a consequence, the cell will not move forward. Okay. And the third kind of motility in 2D, we said is 2D contractile, okay, where your adhesion is required, C is required, but P can be very small. Okay. So, this kind of migration is called a bleb based migration. In 3D, similarly, you can have regular 3D migration, okay, where okay. So, in 3D normally adhesion is not that important, partly because the surrounding matrix can provide you the resistance for the cell to push against it. Okay. So, adhesion can still be small and cell can move. You can have 3D protrusive, okay, where protrusion is major, A is insignificant, C is insignificant, and this is through matrices where the pore size is comparable or bigger than the nucleus, okay, because the nucleus is the stiffest component. Okay. And the third one is 3D contractile, where okay, where you have the formation of blebs, okay, and the nucleus is actively deformed. Okay. So, in 3D contractile you have myosin contractility, squeezing the nucleus. Okay. We will discuss this in greater detail later in the course. Okay. With that, I again get started with the paper we started discussing on adaptive force transmission. Okay. So, what the authors had done was transfected cells with dendritic cells with GFP life act, okay, and monitored the retrograde flow, okay, and using chymographs. So, once again, what is a chymograph? You have a membrane, you draw a very small strip somewhere, okay, and along the length, okay, along the length. So, you have outermost point and this is innermost point. Okay. You track it over time. Okay. So, this is your time axis and this is your length. So, this is your outer point, this is your inner point. So, if you had a fluorescent signal, okay, if you had a fluorescent signal, then what you would have as a consequence of retrograde flow is you will have a slope which you can draw. You can see the intensity which is at certain point here, it goes on getting inside and inside. So, this is the signature of retrograde flow, this slope. Okay. And the slope of this line will give you the exact magnitude of the retrograde flow. Okay. So, when the authors did these experiments on polylysine or adherent condition, okay, what they observed was this. Okay. In control cells, you had a certain magnitude of retrograde flow. When you inhibited polymerization with latranculin, you had comparable value, but when you added latranculin plus pleb, retrograde, so this is my retrograde flow axis. Okay. So, this was completely abolished, okay, suggesting that you have myosin motors once again similar to mesenchymal migration, you have myosin which contributes to retrograde flow. Okay. Now, to check, so this is under adherent conditions. Okay. To check how these cells would migrate under non-adherent conditions, the authors used a setup. Okay. So, in side view, what you have, so this is a non-adherent plate. Okay. They polymerized a layer of agarose. So, this is agarose which is inert. Okay. Agarose is inert. So, the cells cannot adhere to agarose, and what they did then was they punched two holes on two ends of the well. Okay. In one end, okay, in one end they introduced cells, and the other end they introduced chemokine, okay, CCl9, okay, 19, CCl19, which attracts the cells. So, in this setup, so if I zoom in this zone, then what you have is a cell which is migrating under a layer of agarose. Okay. And under the influence of a chemokine, so gradient will be formed. Okay. So, this is my chemokine gradient. Okay. The cells will migrate from left to right, okay, under confinement and non-adherent conditions. So, it is non-adherent and confined, and it is chemotactic migration. Okay. So, under these conditions, when they tracked the retrograde flow, okay, so if I plot my retrograde flow, okay, okay, was negligible. Okay, retrograde flow in control cells was negligible. Okay. Now, what they did was, if you have a cell like this which is migrating in this direction, okay, you can draw two lines. Okay. One is along Y. Okay, this is the retrograde flow that they measured along line 1. What they also did was they did the perpendicular section which is along line 2, and along line 2 they found less amount of retrograde flow. Okay. So, there is negligible retrograde flow. Okay. So, they then transfected cells. So, this was LifeAct GFP. When they transfected with MLC GFP, okay, MLC is myosin light chain tagged with GFP. What they found, okay, was the same cells. Okay, so similar cells, I can draw these cells. Okay, so these cells did not have. So if I were to draw the actin distribution in these previous cells, you would have a flow, a very a prominent actin zone here. Okay, and otherwise you have diffuse zones everywhere. Okay, in the case of myosin, okay, myosin was actually started. This distribution started from far off. Okay, and then all over the place. Okay, so at the leading edge, no myosin at the leading edge. Okay, but when they plotted the retrograde flow, again as before, what you had from the sides, okay, from the sides you had flow inward like this and inward like this, okay, as if there is a contraction from the sides. Okay, along line 2, so along line 2 you have a contraction. Okay. So they then asked, okay, are integrin adhesions required for this kind of migration? Okay. So what they found was integrins, okay, integrins were present. However, integrins were not needed for sustaining confined migration. Okay. So this can be measured, okay, this can be tracked once again by tracking the retrograde flow. And to their surprise, what they found was, so if I draw a chymograph, okay, this is a chymograph in an integrin deficient cell. Okay. So this is the membrane which is moving forward. Okay. So again, once again, this is my time axis, this is my length axis. Okay. What they found was, they found again these streaks indicating retrograde flow, okay, but the magnitude of the retrograde flow increased significantly. Okay. So if this was the average retrograde flow in control cells, in integrin deficient cells, okay, integrin null cells, the retrograde flow was significantly increased. So they then asked whether is it because, so this higher retrograde flow, what about the protrusion rate? Okay. Surprisingly, the protrusion rate okay, between control and integrin null cells was same. Okay. So this is your wild type or control, and this is your integrin null cells. So same protrusion rate. So what this suggests is that when you remove the integrins, overall the actin polymerization rate. So what is actin polymerization rate? I can write down the actin polymerization rate okay, equal to protrusion rate plus retrograde flow. Okay. So if you do this, then what you would find is actin polymerization rate increases in integrin null cells. Okay. So this is an adaptive response. Okay. And what the authors then went on to show that any weakening of the ECM cytoskeletal linkage, so of ECM cytoskeletal linkage led to increased actin polymerization. Okay. So this they proved by doing experiments with talin negative cells, okay, talin null cells, and by plating cells on PEG. Okay. So PEG being inert, cells cannot form adhesions. Okay. However, there was one important difference. Okay. So first of all, okay, two things to be said. One is, if your overall actin polymerization rate, okay, in talin null cells was this much, okay, Okay. In PEG, the actin polymerization rate was not to the same extent. Okay. So, this difference, there was a difference, okay, between tailin null and on PEG surfaces, okay, and this difference can be partly attributed. So, first of all, there are two questions, okay, that how does retrograde flow increased. Okay. So, one possible reasons, okay, so how is it that you can have retrograde flow to increase in integrin null or tailin null or on cells cultured on PEG surfaces. Okay. So, what there is a possibility, okay, that since, so if we assume that membrane tension is constant, so if we assume that membrane tension is constant, okay, so that means that to maintain, if this were to be constant, now in case of higher retrograde flow, so increased retrograde flow would decrease the membrane tension. So, how can you maintain the membrane tension cost? By adding more amount of monomers or by increasing the rate of polymerization. Okay. So, for this to be constant, since retrograde flow is high, for this to be constant, what you can do is, okay, you can have higher incorporation of G actin into filaments. Okay. Okay. So, thus far two things we found for dendritic cells. So, under adherent conditions, you have retrograde flow. Under non-adherent conditions, you have no retrograde flow. Okay. But when you, in case of integrin null cells, okay, retrograde flow is very high. Okay. So, the authors next asked that how fast, so if you were to subject the cells to a different situation, so for example, let us say this is your background of PEG surface, and on this PEG surface you randomly draw these lines of serum coating. So, this is serum coating, okay, or adherent. Okay. So, your background is non-adherent. So, this area is non-adherent, but this is the only adherent stripes that your cell sees. In these cases, is the cell able to change its cell speed or adapt its cell speed in real time? Okay. So, what they tracked was the cell speed, okay, the average cell speed as a function of time. Okay. And so, if I were to draw these zones, let us say this is one zone in which you have an adherent spot, this is another zone where you have the adherent stripe. Okay. And what they found was when the cell was migrating on these zones, your cell migration rate or cell speed was unchanged. Okay. So, your cell speed remained unchanged. So, even if you provide the circumstance of adherent and non-adherent zones mixed together, the cells can adapt that to it in real time. Okay. When they did the same experiment with fibroblasts, okay, again you have this background and you have these red stripes, okay, and you did the experiment with fibroblasts. Okay. What they found was there was retrograde flow in adhesive zones, but no movement. Okay. So, how would the chymograph look for this kind of a situation? Okay. So, let us assume if I were to zoom in on a cell, okay, let us say this is a cell, okay, and let us say this edge, okay, is a adherent spot and the background is non-adherent. So, this is a cell. Okay. So, if you plot the chymograph, what you will find is there is a zone in which you have retrograde flow, but in the other zones your retrograde flow is flat. Okay. So, this is my again time axis. Okay. So, this is your adherent zone and this is non-adherent zone. Okay. So, your retrograde flow is 0 in the non-adherent zone, but it is positive in the adherent zone. So, which suggests that the difference between fibroblast and these dendritic cells is, for a dendritic cell, let us say, okay, let us assume this is an adherent zone, okay, and this is an adherent zone. Okay, this is adherent. Okay. So, what the cell does is the retrograde flow is very negligible in an adherent zone, but what the cell does in the non-adherent zone, the retrograde flow increases. Okay. So, this is for a dendritic cell. Versus for a fibroblast, this kind of accommodation is not possible. Okay. So, if I were to draw a picture similar to that of a molecular clutch, okay, what you have is as possible. Okay, like before. Okay. So, this is in an engaged state. Okay, you have two things. Okay. So, this is the protrusion and this is the retrograde flow. Okay. What I have drawn here is the filament and this is the integrin connection. Okay. So, when you have a situation, a slipping kind, what is a slipping kind? Like you observe in PEG coated surface, where your integrin is there, but it is not engaged to the surface. So, you have this filament, your retrograde flow is partially increased, but your protrusion remains the same. Okay. So, your retrograde flow is partially increased. In the other case, you can have uncoupled. What is uncoupled? When there is integrin not present. Okay. In this case, what the cell does is it increases retrograde flow and its protrusion rate, okay, is this. Okay. So, as a consequence, what you find is its protrusion rate is always maintained constant, okay, but the retrograde flow, so actually these lengths should be the same. The magnitude of the retrograde flow is extended. So, retrograde flow increases under independent conditions. Okay, and it is set by the extent of coupling. Decides okay. So, with that I end our discussion on amoeboidal migration. Okay. So, you will have another reading assignment. This is the Renkawitz et al. Paper. Nature Cell Biology 2010. Okay. So, I will just touch upon another type of migration. So, we have mesenchymal, we had amoeboidal. Okay. So, in both these cases, the cells migrate independently. Okay. Or in other words, you can say this is an example of single cell migration. So, just you understand that if you were to think of wound healing, okay, so what do you have? You have groups of cells which migrate. Okay. In the case of wound healing, instead of single cell migration, what you have is collective cell migration. Okay. So, by collective cell migration, okay, you have a group of cells. So, let us say this is a group of cells. So, each of these is a cell which is migrating, okay. But it is not necessary. So, the way I have drawn this, that cells are sticking to each other. So, you can also have a situation when you have a group of cells migrating, okay. All of them are effectively migrating in the same direction, but there is some other signaling between each of these cells such that their relative distance is maintained. Okay. With that, I stop here for today. In the next class, we will discuss about collective cell migration. Thank you for your attention.